on your body. So examples, garlic, green tea, tomato, pancreas, all these are rich in secondary metabolites or natural products, and they exert a health benefit in your body. And I specialize in the analysis of functional food using metabolomics. So at first, let's hear about metabolomics from a cartoon made by University of Finland for 3 minutes.
Jarmo is a Finn, and like a good Finn, he loves trees. He loves them in the forests. He loves them in the xylitol gum he eats after every meal. And he loves them while he is in the sauna smacking himself with Vasta. He loves them so much, in fact, that he has made a career of researching trees. One day, on his rounds of the trees in his laboratory, Jarmo noticed something wrong with some of them. He tried every routine test to figure out what was wrong, but his trees kept getting worse. Jarmo did not know what to do as he watched his beloved trees withering away. Jarmo became depressed, which is relatively rare for a Finn, at least in summer. One day, Antti, Jarmo's colleague, told him about metabolomics, a field of study where researchers can break down samples into their smallest components and assess them in great detail. More specifically, metabolomics is the analysis of cellular processes based on the chemical traces that they have left behind. This analysis then creates a picture of the differences between sick and healthy trees. This can be useful in food production when establishing the chemical makeup of a food or controlling for quality between different rounds of production. It can be used in medical research to help understand the progression of diseases and to assess the impact of a person's diet and metabolism. Jarmo saw that through metabolomics, he could now get a window into what was or was not happening correctly in his trees on the most fundamental cellular level. Now, he might finally be able to figure out what was going wrong in his research.
So now that you have listened to Jarmo speaking about metabolomics and hope you have a good idea about the technology, now you will listen from myself. So again, as I mentioned before, we are going for the natural product, or what we call the phytonutrients, and there are so many classes worldwide in different places in the world, different plants, and these are some major examples. And our job as pharmacists or phytochemists or natural product chemists is to identify the active agent in the drug or in the functional food. It goes by folk medicine evidence, then we go the classical way, the isolation and the biological evaluation. And it's a long path, as we all know. One example is well known, and we know the evidence, is coffee, that I drink a lot. It's my favorite beverage. So we drink coffee because we knew that caffeine is inside coffee and it's behind the CNS effect. But in many other cases, we don't know the exact chemical or chemicals. To expedite the discovery of the active agent, this is where metabolomics can pop in, and it helps to identify the active agent in a rapid way, not the classical path that we used to do years ago, in some sense. Not always successful, but it helps in several examples, as I will show you today. So identification of the active agent is expedited using metabolomics. So it's all about the bioinformatics, and as I call it, it's a marriage, a wedding between computer science and molecular biology. It helps us to expedite the discovery. We typically use analytical platform, MS and NMR, generating huge data set. To help identify the active agent, we use the bioinformatic tools. Slides are very simple. As I always tell my students, everyone has data, but the smart scientist is the one who can extrapolate the information from the data. And this is what bioinformatics can help you in that regard. Two of the most common tools that we use a lot is PCA and PLS. The application, we do it for better plants and better humans or better disease treatment. We do human studies. but today I will not touch upon. It's mostly only the food application. These are the questions that we ask in my lab, the metabolite profiling, the quality of the food, the detection of adulteration, and the authentication of the food. Coffee, again, is my favorite beverage, as I've mentioned before, and I think it is the one that we have investigated the most in my lifetime. So we simply ask questions about roasting of coffee using NMR and mass spec. We used LCMS to assess the effect of fermentation in coffee. This is the famous animal in Indonesia. It's called the luwak coffee, one of the expensive coffee products. And we look at the effect of coffee fermentation by the luwak on its chemicals. We started to look as well at different coffee varieties. We have the coffee Arabica, the coffee Robusta. We start to look at gut interaction, and I'll tell you one example today. How does our gut microbiota interact with the coffee chemicals? We knew that coffee and tea can help in reducing weight, and so we compare different type of coffee and tea, black, green, fermented, unfermented, to see how each would affect with a battle obesity problem inside your body. It's a whole story, and I give this quote. It's my story about a beverage we drink. Nothing else can make us better think. So it's gonna be like a movie today, a morning movie. Today, right now it's 10:30 at Cairo, and I'm the producer of the movie, and these are the heroes that you will see telling you the story. The reason behind such type of presentation, maybe you haven't ever attended to my lectures, is that I believe a lot in less is more. We don't need to overwhelm our audience with too many details and slides. Better simple to deliver the message. And this you should be, I think, as well in teaching nowadays for our students. Infusing real life experience is also important in any talk. I think a couple of years ago, I gave a talk about giving talks at the comstech. And I think when we infuse life experience in our talk, it delivers the message more. It's a challenge, but we try to accept the challenge. Again, I will not speak for 1 hour, 30 minutes, 1 hour is good enough. And you are online, so instead of losing you and to keep you attentive, I will try to be as simple as possible. It's gonna be told by, I know it's a bit, I take a scene from a famous movie, and today the take-home message will be taken from the Cinderella story of metabolomics. So metabolomics at the beginning was underestimated as a technology, and like in the Cinderella story as a chemical, it escaped the scene, but later on, it proved to be powerful and of potential. Of course, we have to admit that the discovery of metabolomics and it happened after the post-genomic era, after the discovery of DNA happened in 1952. It paved the way to all omics technology, to be honest, although chemicals were well known centuries before genes, as we all know. But accidentally, it was the discovery of the gene that opened the door to all omics sciences. It happened in 1953, led by the great scientist Watson and Crick. 2001 was a remarkable year in human mankind, witnessing the completion of the human genome that was published in Nature and Science. And at that time, I wasn't that much involved in science, but I was a PhD student anyhow. Maybe with people, scientists and researchers expected a better outcome, meaning that we know the secrets of the genome, we know the code, we would know how to treat disease, how to prevent disease, but it's not the case anymore. And we know that we are still challenged by other factors. Lately, 3 years ago, this article came out in Time saying why your DNA is not your destiny, because there are other players. The other players, I take you to Cairo, and this is a very old district in Cairo called Khan al-Khalili, if you happen to have visited. And you can see people walking in the street of Cairo, but you can never tell what they're doing in their life, what are their jobs. Because simply, as I always say that they are silent. They are not speaking at the moment. Like genes. Genes are silent on their own. And when do genes speak? The genes speak when they deliver their product, the chemical. And metabolite, as we all know, is the end product of a gene expression. So we own a gene, mRNA, protein, and chemical. So per se, in the middle, you can see this person selling water tank. So you can tell that this is his job. He's a waterman. The same happens anywhere in China, in Egypt, in India, in Pakistan. People rumbling in the street, you can never tell what they are doing because they are silent at the moment, like most of you in this talk. And this is what I call the life iceberg. And this is the famous Titanic ship. And we all know the story that Titanic. Now you're all awake, I hope. It happens in science as well, the science iceberg, meaning that in any science process, in any question we are asking, we can target one aspect, ignoring other aspects. So maybe as a molecular biologist, would be looking at the gene level, ignoring what's happening inside the cell, the protein and the metabolite inside. And this is what brings us now to what is called as system biology, where we see an integration of interdisciplinary or a sort of an interdisciplinary research. I am, of course, in favor of metabolomics, and this is why I call it the canary of the genome, the speak out of the genome. Cinderella story. The Cinderella is eating her food. What would happen inside her body after eating her meal? Nothing would happen at the gene level, very little. Protein, less affected, but all the changes are happening at the metabolite level. And as I always think of it, chemicals define our phenotype as this example. These are the application of the omics, of course, aging, disease, drug discovery, nutrition, lifestyle. We use it to answer these questions. So if till now you don't know simply what is metabolomics, I will put it in that simple. As I mentioned, chemicals define your phenotype. If you happen to be diabetic and you are measuring your glucose on a daily basis, because glucose is a marker of diabetes, this is what we call as a single metabolite analysis. But if you are measuring the hundreds and thousands of chemicals inside your body, this is what we call a metabolomic analysis. And if you are relating the metabolite level to the gene expression, this is what we call the functional genomics. Simply, what you see in front of you is a tree, but if you zoom on the tree, you see a very different scenario. People standing. And this is exactly what happens in analysis. We are zooming the level of detection of chemicals in metabolomics. It's not easy, to be honest. It's very challenging, and we are very uncertain of all the identification we make, especially for the minor compounds. It's exactly like your eye vision test. You can see your gene very with high acuity, with confidence, because of many reasons. I don't want to go in details, but chemicals are unfortunately very, very difficult to be seen that much accurate. So if you see your gene 6 out of 6, your chemical is seen 6 out of 60. And this is the big challenge in any metabolomic experiment. On the other hand, people working with genes, they are playing with 20,000 genes per se, and I put it as such as like your money in your bank account. You don't feel it on a daily basis. It's what shape your life is your what you spend on a daily basis inside your pocket. And these are the chemicals. They are less, but they are influential. They're shaping your daily life. Again, the problem of drawing the picture of the metabolome using different technique, the problem that we don't have one technique with which we can get the picture. I'm a painter in my younger days, but I quit it anyhow. You can see this is the painting of the Mona Lisa. If we painted using NMR, you can see it, but of low resolution. On the other hand, I'm sorry, MS delivered this picture, but it doesn't capture all the details. Until now, we don't have a technology with which we can capture the Mona Lisa in the right way. I was traveling to Turkey last year, and I thought of the metabolome like this flight. It's and when you take a flight, the truck pull the flight exactly like the analytics, but it cannot fly the aeroplane. It would fail in that regard because simply the metabolome is bigger than the analytics. This is my metaphor. Maybe I'm wrong and you disagree, but this is what I think over these years. We are not pulling the metabolome to fly till now. The workflow is simple. We have a sample. It can be a plant, a human, a bacteria, an animal. We extract the sample. We do the detection, identification, and then we do the multivariate data analysis. As I mentioned before, it's all about the bioinformatics as a marriage between computer science and molecular biology. Now I will tell you 4 stories in different fields to show you how metabolomics can help in food science. Let's start with the first one. Phytoestrogen, functional genomics. It was my first experience in metabolomics. This is why I always prefer to. So phytoestrogens are chemicals present in legumes and only in legumes, and they are plant natural products that exhibit an estrogen-like effect. And this is why we call them as phytoestrogens. They are very famous in the Far East, especially because of the consumption of the soybean, as we all know. And they do have a function in the legume plants. They are behind the symbiotic relationship between the rhizobia to fix nitrogen. Imagine. And the question I was asked at that time is to draw the pathway for the production of the phytoestrogens. It's like a pizza. We all like pizza, but when we look for the authentic pizza, we might go to Italy to enjoy it or to savor the taste. Because we don't know the exact ingredient, we don't know the exact recipe. And this is what we are looking for in biosynthesis, to know the exact recipe of the production of these phytochemicals. So to know the recipe and to remember the first slide that I mentioned that genes are silent. So to make genes stressed and to make them produce these chemicals, as we all know that natural products are almost produced as a defense response under stress, we started to elicit the plant. It was a cell culture using different elicitors. One elicitor was yeast to mimic infection. The other was jasmonic acid, a phytohormone. After that, we collect sample, analyze them at the gene transcript level, not by myself, by a molecular biologist, and I was in charge of the chemical analysis to draw a biosynthetic pathway for the isoflavonoid. Just to tell you that it's like a puzzle that we assemble. Although that the pathway was drawn before, we managed to identify novel natural product and novel enzymes in the production of the isoflavonoids using this technique. Can we identify using such a large-scale experiment new natural product, new effects, new uses? So we identified this chemical called haspidol, and this chemical exhibited an antifungal effect. We tested against a fungi in a petri dish, and you can see clearly that the haspidol can restrict the growth of the fungi compared to the solvent. So it has an antifungal effect. And looking at this novel chemical, who is behind, we went to our microarray ship and looking at literature, it's believed to be produced by peroxidases, which is a big gene family in legumes, a huge gene family. So we had 90 candidates that might be involved in the production of the haspidol. A big experiment to clone 90 gene is a long time and effort and money as well. So at that time, we looked at it in a nice way to narrow down the candidate that we need to clone. So we decided that we do have a large data set. We look at comparative elicitors. So what was really nice that this chemical, the haspidol, was inhibited by jasmonic acid to the left and induced by yeast. So we decided to look at only the genes that were induced by yeast and inhibited by methyljasmonate, kind of identifying the criminal in producing this chemical. So when we did this comparison, we narrowed down we did identify the guilty genes. So from 90, they went to 3 candidates. It's exactly as I always think of it, differences tell the story. And you all know that carbon and diamond are the same. It's just a different way of assembly. So doing this way, this function comparison between the gene and the metabolite, we cloned only 3 instead of 90. So we managed to save both time and money. So in summary, for our first story, we identified new natural product, mechanism of elicitation, a new chemical, the haspidol, and the peroxidase involved in haspidol production. Do phytoestrogens stop the stroke? Meaning that if I'm an Egyptian, decided to consume soy milk on a daily basis to benefit from the phytoestrogen, will it help me? The answer is sadly no. It's very debatable. It's called, by the way, the Asian paradox, if any phytochemist is among us. So among these ladies who would benefit from the phytoestrogen, it is most probably the Asian baby.
I am the one who can benefit from the phytoestrogens.
This is my voice, by the way, of course, using AI. Why is that? Our second story is gut microbiota. Asians or Chinese people, due to lifestyle and genetics, they harbor inside their body the microbe or the gut bacteria that convert the phytoestrogen to the active form. Egyptians, as an example, might be not the right example, but anyhow, we don't harbor inside our body this microbe. So we don't convert the phytoestrogen to the active form, that is the equal. So this brings us to what I've been interested in the last 10 years, is food-gut interaction. It's still a new field. We are still exploring. It's a dark side that we are trying to understand using metabolomics. Bees, gut microbiota is a very interesting living organism as well, if one would like to consider his career. The bee has a very interesting story in their gut microbiota. Anyhow, it's a dark side that I have decided to venture. So to give you a quick hint, inside your body, you have the good bacteria and the opportunistic bacteria inside your gut. And when you pay attention to these microbes and eat the good food, the rich fibers, fruits, and etc., you flourish your good bacteria and good function inside your gut. But if you go to the processed food and this brand, famous brand and whatsoever similar, you get more of I don't call them the pathogen bacteria, but rather the opportunistic bacteria. They take opportunity of you when you are weak. So these are the function of the gut microbiota, not only in digestion, they produce vitamins, they protect you from pathogen, many functions. And most phytochemists know that we all know that the phytochemicals in food, most of them, they have poor bioavailability and they are not converted much inside your gut, your digestive system, except when they reach the colon at the end. And this example is pomegranate, a famous fruit in Egypt and many places. This is a very complex phenolic in pomegranate called punicalagin. It's a polymer, and nothing happened to punicalagin when you eat in your GIT, except inside your colon. Under the action of these microbe, it gets converted into these small molecules, urolysin, and then they get absorbed. So this is known. I'm not claiming any. What I decided to look at in a different way, because I'm a coffee addict and you don't see my coffee mug, I'll take a sip. Anyhow, so I wanted to look at what happened to the gut microbiota own metabolism when you drink coffee on a daily basis. So after drinking coffee inside your GIT, what happens to the gut microbiota metabolism itself? So we did that, not in a human, but in a culture. So we exposed gut culture to different functional food, including coffee. We could see that when we add these coffee extract, we get the short chain fatty acid, which is the energy molecules needed by the bacteria, which is known. But what was very interesting that we observed an increase in GABA. GABA is a CNS depressant inside your body, and it's kind of that the gut microbiota is not happy with the production of caffeine. I'm putting it in a funny way, producing this CNS depressant. Think sadly, someone marked on the screen, I would have to log out, but this would take time. So anyhow, this was published in the Journal of Advanced Research, a leading journal nowadays. It just came out to be now the 6th among multidisciplinary field, and I'm the acting editor-in-chief. And just to tell you that this paper, I think, was a good paper. It got 25 citations in the first year. And I would like to welcome you all and consider you any submission from your group. We have recently started a new journal. I'm the editor-in-chief called Science and Technology Nexus, and you can reach me out if you're interested in a submission. It's a free journal at the moment, and it's not a multidisciplinary, but an interdisciplinary scientific journal. Just came out 2 weeks ago. I think this is a cartoon I made for the judging of papers. I gave a talk to Comstech before, and this is a science nexus. So the second thing that we looked at with gut microbiota, we always claim that polyphenols are antioxidants, which is true. What about if we challenge them with the oxidant inside your body with the absence and presence of gut microbiota? For example, how would the polyphenol in green tea or in pomegranate interact with a pro-oxidant in the presence of a gut microbiota versus the absence? Is it the same scenario, or gut microbiota is playing a role in the antioxidant effect of polyphenols? To test that, we did an in vitro culture where we exposed gut bacteria to the oxidant and the antioxidant, and we analyzed using LC-MS. Interestingly, in the absence of the gut bacteria, we did see a production of the quinones. In the presence of the gut microbiota, these quinones were quenched. So it's a hint that we can say that the gut microbiota can modulate the antioxidant effect and protect your body from the oxidative stress. We don't know the bacteria that did that, but we knew that this is evidenced. So inside your body, I put this scenario from the Titanic. Fusobacteria is a gut bacteria that is believed to be involved in colon metastasis. And this could be one scene of Titanic where you see the officer shooting at the passenger in a bad relationship. But on the other side, which is more dominant, you see a positive relationship between the two heroes surviving for each other's life. And your gut microbiota would not leave your colon and leave this relationship for the benefit of both sides. So, conclusion from Titanic. Remember when the lady said,
"But now you know there wasn't."
These are all the reactions that happen inside your colon, very complex reactions when you consume your food or when you're under any stress happening by the gut microbiota.
But now you know there was a man named Jack Dawson, and that he saved me in every way that a person can be saved.
So if the colon were to speak, would say, now you know there was a bacteria I don't know that saved me in every way a colon can be saved from an oxidant. And the conclusion, I put it as such, think of your gut guest in the food you would waste. And if you love your bacteria, when you go to your restaurant, look at the menu and what pleases your gut microbiota. Read about that. So from humans to plants, another story involving bacteria, but not living inside human, but inside the plant. And this is my favorite story I will tell you today. Better plant growth. Typically, opposite to plant, we all know as phytochemists or food chemists that plant aroma or food aroma are nice. We enjoy the aroma of peppermint, of citrus, but bacterial aroma tend to be offending. We dislike the aroma of bacteria, and you can tell it. When a food is spoiled, you can tell that the food was spoiled because you can recognize from the aroma. But believe it or not, some of these aroma chemicals, they have a good effect on the plant and humans as well. Let me tell you from this discovery, an old one dating from my PhD study. So what you see in front of you is a petri dish. And on one side, there is a plant growing neighboring a bacteria. And on the other side, a plant is growing without any bacteria. But in both cases, there is a wall in between. So the plant is not in physical contact with the bacteria. They are distant from each other. And what you can see that the plant growing neighbor a bacteria with a wall, there is a cross promotion effect. The plant is growing bigger in size. And the only explanation, if anyone can anyone think of what would be the explanation? Write it on the chat box. Anyone can, to see what is your thinking? What is happening here? 1 minute. A chemical of what type is produced by the bacteria? Anyone can guess? The nature? Don't want to take too much time. Okay, it's a volatile. And this volatile is like a perfume. So the plant here is smelling the perfume by the bacteria and responding by increasing in size. My job was to identify this chemical produced by the bacteria or to resolve the bacterial perfume using GCMS. Came out to be, and again, remember I told you, bacterial aroma is very complex and not easy to resolve. So to fish it out, again, I did this comparison. So I knew that there are strains that are active and strains that are inactive. So I ran them in parallel. And doing that, we narrowed down the peaks to these 2 peaks. And they came out to be very simple chemicals that we took in chemistry, 2,3-butanediol and acetone. And without going into too much details, they were confirmed to be the active agent behind the growth promotion effect. So again, differences. This is the aroma of a bacteria, believe it or not. And this is how metabolomics, you can see around 20 peaks or 30, but when we ran it on the GCMS, it was only 4 peaks. And this can tell you the power of metabolomics in resolving the complexity of the aroma. So conclusion, soil microbes can present a source of agrochemical. In 2017, we published the protocol in Nature Protocol, and it was placed on the cover page. So again, conclusion from Titanic.
But now you know there was a man named Jack Dawson, and that he saved me in every way that a person can be saved.
So if the plant were to speak, would say, now you know there was a bacteria that helped me in every way a plant can grow better. Now I will have to but again, a paper is just a fruit unripe. If you apply it to the society, you enjoy more the flavor. This is what we do. Much more is the analysis of nutraceutical in my lab. So we look at the quality of a nutraceutical, what chemicals makes a health effect, and the effect of processing, be it roasting, be it drying, be it fermentation, and all of that we do using metabolomics. We did it at first in collaboration with the Alexander von Humboldt. They supported me at my junior life after getting my PhD. It's a great foundation, and I encourage anyone to consider after earning your PhD. So the quality control of a function, now I think I will have to log off because otherwise, or unshare, 1 second, stop shares. Good. So quality control in functional food is not easy as chemicals because of so many variation due to species type, the variety, the cultivar, where the plant was grown, how it was processed, and the post-harvesting processes. To give you a quick hint, so per se, you want to eat an apple among 4 apples. You would judge it by the red color as one marker. But an apple is not only a red color, it's much more inside. And this is what we call the targeted way. So if you're only measuring the red color abundance in the apple, this is what we call a targeted analysis. It's fine, but this is what the approach. If you're looking at all the phytochemicals in the apple, this is what we call the omics in quality control. And I put it as such, a question, which one is more challenging, analysis of an apple or a tablet of aspirin. It's certainly, apple is more challenging due to the complexity inside. And as a food chemist, I would say, eat it and don't drink it, because a juice of a fruit is just sugar, but to benefit from all the phytonutrients and the fiber, eat it as a apple. These are the drugs that we have visited. Many were Egyptians and many were German. Licorice was one example. I will go quickly. So in licorice, we collected different genotypes from Africa, Asia, and Europe. And we want to know which one is the best. So are Asian people having a better source of licorice than those living in Europe or in Egypt? So we analyzed them using NMR and mass spec, as I told you before. And this is what's called a PCA, simply to classify the different licorice types. And you can only see, I don't want to explain the PCA principle, but it's simply the separation of samples. They are not similar. We don't see similar results. And this is why we opt to the comparative approach, meaning, and even this can apply in anything, as I always tell my students, if you want to prove your hypothesis, try to test more than one technology or technique to be sure of the results. Because here you can see that NMR to the right is giving a different PCA plot from LCMS to the left. Simply because, as I mentioned before, each technique has different detection principle. 2D NMR is a very powerful technique in structural elucidation, and we all know that. I started to enjoy using 2D NMR to identify food chemical in the crude extract without any sort of separation, relying on the beauty of the 2D NMR experiment, these complex spectra. It can help identify complex chemicals. So in hops, which is a famous plant in Germany, they use it in brewery, in beer. We identified this component in the complex extract. And interestingly, it was opposite to what we always say. It was a degradation product. So if you want to get more sleeping effect in your hops, kill it more, because you regenerate more of the active sleeping agent. So not always it's better to keep cold. This was a nice coincidence where we identified in Egypt an old brewery in Upper Egypt producing beer. And they have all the evidence of the beer making, and they wanted to analyze the beer itself to prove it was a beer. It was a very old brewery in size and age. So they give me this residue after 4,000 years. Of course, there is no any beer. It's a solid, it's an ash. But we managed to identify that this is a beer. I call it a recipe of an ancient pizza that it was a challenge to identify. The most interesting result that we had that the ancient Egyptian, they knew about flavors because different lines in the factory had different profile. And we identified that the preservative was a type of phosphoric acid, believing, of course, we are not sure how it came inside and the source, but at least that phosphoric acid that we consume in our cola was known to the old Egyptians as well. Metabolomics can help in identification of many post-genomic and pre-genomic, prehistoric era. The secrets of mummification is one thing. There was a paper, it's not mine, that resolved the mummification process using metabolomics, published by colleagues of us in Egypt. The last thing that we look at is the effect of processing, cold versus hot, and how does the preparation of a food affect its chemical composition and health effect. This was done using roselle, a famous flower in Egypt. And in roselle, there is a belief that if you drink it hot, it can lower your blood pressure. It can increase your blood pressure, sorry. But if you drink it cold, it lowers your blood pressure. So we wanted to test this hypothesis, and simply by preparing roselle using different method, cold method and hot method, and analyzing Roselle using NMR. Simply what we see that Roselle that is prepared using hot water has more of the hydroxycitric acid, which is a slimming agent, not like the these now the I forgot the name, Ozempic and all these things using to lose weight. This compound is famous in grapefruit and it helps you to reduce weight. But if you prepare Roselle using a cold method, you get more benefit of the anthocyanin that lower your blood pressure. Published around 7 years ago in food chemistry. Carob is another plant that we look at grown in the Mediterranean region. And we look in carob at the effect of roasting and ripening as well. Carob can be produced in green, unripe, or ripe stage. So the question we ask in carob are the ripening effect as it goes from green to brown. The origin, we collect carob from different places in Egypt. And the roasting, because before eating carob, you have to roast it on fire. It induces the Miller and the caramelization of sugars to improve the taste. These are the places from where we collect carob in Egypt, Alexandria, Oasis, and in Cairo, Giza. One of the most challenging questions in for any food chemist is the identification of the active agent, which I often give it as an example, finding the needle in a haystack. This is the last example of a date grown in Egypt. And we all know that date fruits shouldn't be consumed by diabetic people because of their high sugar level. Except this exception is a date that is produced in Egypt, in Upper Egypt, called Balanitas date. It doesn't have that much intense sweet taste. It's both sweet and bitter. And bitter is behind the reason why it lowers the blood sugar level. And again, comparison. This is the last example I put. So to identify the active agent, it came to my mind it wouldn't be easy. So I decided to analyze this date along the edible date, the known date that we consume in Ramadan. As you all know, Middle Eastern, and I'm not sure in Pakistan, but we like to break our fast on date because date are very rich in sugars. To break your fasting. So anyhow, I prepare an extract of the date, that the sweet date and our special date using NMR, and both are rich in sugars. But when we look at the aromatic region, we identify in our Balanitas date these signals that came out to be matching exactly to a very famous alkaloid and a very potential anti-diabetic drug called trigonelline. Trigonelline is an alkaloid present in fenugreek, and it exhibits a very potential anti-diabetic action. And this is one experiment. So there was a project funded by the EU to identify it, and they didn't reach out because they didn't follow they followed the classical way. And this was just one experiment with which we proved for the identification of the anti-diabetic agent. It was published in Pharmaceutical and Biomedical Analysis. So doing the classical phytochemistry, sometimes you can lose your goal. But can metabolomics be like the Suez Canal, a shortcut? So instead of reaching to Pakistan, long ago we had to go around the Horn of Africa, but now with the Suez Canal, it can expedite the reaching. I wouldn't go to conclusion from the Cinderella story. So as you have seen earlier, metabolomics escaped the scene of food science, but it could be the right match in size for the Cinderella story and the fit of the story in system biology. But to be honest, it's the orchestration of all sciences, metabolomics, transcriptomics, genomics, and proteomics, all together when well orchestrated like this symphony. So opposite what I said before, metabolite define the phenotype. It is the orchestration of all omics. Thanks go to my lab members and people who contributed to the work, and they are the heroes behind it, and to the funding and the support from the Alexander von Humboldt, particularly in Germany. And this is my life. I would like to tell you as someone living in what's termed out a developing world or countries with limited resources or whatever. As I always put it, this is a cartoon not made by myself, but a student of mine, showing my life like any scientist. But what I would like to tell you that in Germany or in US, when I lived at the old time, I had a fast car, but the car wouldn't fit the road in Egypt, the bumpy road. And it was only the bike that I managed to use and to reach out. This is my advice. And this is Cairo, Egypt. This is if you have time, let me show you. This is the movie.
I'm an artist at my heart,
Explaining all what we do.
and ever since I was a kid, I was inspired by nature. As I grew up in Egypt, I decided to study nature more scientifically, and now I've developed this technology called metabolomics, with which we picture living organisms using advanced analytics.
I will share again last time.
And not by the paint and the brush. And this is the career I have developed over the past 20 years. My seminal discovery, actually, although that I work in medicine, it was an agricultural discovery. Opposite to humans, so when you put a perfume on your body, you just smell good. But in plants, when they smell the perfume from certain soil bacteria, what we identified was that these plants start to grow bigger in size and to perform better and be more resistant to infection. So my job was again to identify this chemical that by which the plant is communicating with the bacteria and to resolve the perfume of the bacteria. Came out to be at the end as well that this perfume produced by the bacteria has a role in bacterial resistance to antibiotics. So it's a study that emerged as an agricultural application and winded up to be at the end a medicinal added value. We simply try to understand how organisms function in life by capturing all the chemical reactions that happen inside the cell. By capturing such detailed information, not only that we understand how the organism lives, but rather we can exploit such information to improve the function and to predict whether a problem can happen in the future. So to give you an example, we can better understand how plants adapt to the environment. We improve such adaptation. We can understand why corals are almost extinct at the moment.
So don't want to take the whole movie. This was the end. And please let me show you in 2 minutes. I plan to host another webinar by the publisher. And if any is interested, you can email me or send me a message on scientific writing and consider Science and Technology Nexus, our new baby journal. And in 2 minutes, I will just show you if the screen sharing. Now it's closed. Oh my, Fazima, I can.
Okay, Professor, let me see. I think you should be able to share your screen.
1 second.
Yeah, we can see your screen.
Evolving world, the answers to our biggest challenges don't lie within a single discipline. They live at the intersection of many. Welcome to Science and Technology Nexus, a groundbreaking new journal dedicated to interdisciplinary research that drives innovation across borders and disciplines. At Science and Technology Nexus, we believe that real progress happens when ideas collide, when a physicist's insight fuels a breakthrough in medicine, or when agricultural science merges with artificial intelligence to solve global food insecurity. Science and Technology Nexus integrates insights from multiple fields to address complex scientific challenges. We welcome submissions with a special focus on interdisciplinary research that integrates insights from multiple fields. The journal welcomes research papers, review papers, technology protocols, and case reports. Our scope is intentionally broad and boldly integrative. We cover natural sciences, engineering and technology, medicine and pharmacy, environmental and agricultural sciences, all under one collaborative umbrella. What truly sets Science and Technology Nexus apart is our mission: to bridge the gaps between fields, foster collaboration, and encourage research that offers holistic, real-world solutions. We're not just publishing studies, we're building a global conversation, a network of thought leaders, innovators, and researchers working together to rethink what's possible. From climate change to public health, from sustainable technologies to next-generation materials, with a strong commitment to open access, we're ensuring that this knowledge isn't locked away. This is research meant to be read, shared, and acted upon. Our editorial board brings together diverse voices from around the world, scholars who champion interdisciplinary thinking and ensure that Science and Technology Nexus reflects a global perspective. The journal is published by an international scientific publisher and includes distinguished editorial board members from worldwide, led by editors-in-chief Dr. Mohammed A. Farag and Dr. Haydar Al-Kafagi. In a world that often works in silos, Science and Technology Nexus is a call to connect, collaborate, and create. A platform for the ideas that will shape tomorrow. Science and Technology Nexus. Connecting disciplines, creating solutions, shaping the future.
Thank you, Kazima, and all our attendee and audience. I'm open for any questions you
Thank
have.
you very much, Professor Farag, for an excellent presentation. And I really enjoyed your presentation. And I think presence of 126 people online says the same for you. And you
Thank
can
you.
see, yeah, you can see number of, compliments for your presentation in the chat box. So with your permission, I see some questions in the chat box. I'll take a few of them.
Can you read them, or I will have to go through?
Yes, I will read them for you. I'm just trying to find. Okay. So one question that I have received is from Ms. Lailatul Qadar. She's asking, can GCMS be used as a better diagnostic tool for metabolomic profile of human body fluids like serum, plasma?
Good question. This is a very challenging question. Of course, you have to be, you need to know what you are targeting. I would favor GCMS a lot, especially in our world, because it's simple, more robust, and not expensive. But this is in case that your answer will be lying in the low molecular weight, small molecules, the primary metabolites. But if your question is per se looking at the complex lipids, at the glycoproteins, so definitely GCMS will not be your answer. Then you would have to switch to LCMS. But I would definitely start with GCMS and the preparation of a sample for GCMS, you would precipitate all your proteins at first. So you would never see them anyhow. But again, GCMS is powerful and it's very well suited for for human metabolism. Remember as well, human metabolism is more similar, not like plants. So GCMS is a good option, but again, it's very limited in regard of looking at only the low molecular weight compounds or volatiles. We had one study in very brief looking at heart myocardial infarction, and the marker was hydrogen sulfide, a gas. And only GC detected it because it was a nice coincidence. But again, you have to acknowledge the limitation, my friend. Hope I answered it.
Yeah, thank you very much, Professor. So before I take this question, Professor Farag, I'll request you to please send me email containing information about the Food and Science Nexus journal for which you are...
One second. This is my email, and I will send the link here. 1 minute. And if anyone of the Batibit Junior would like to join the editorial board, just email me and we'd be very happy to include.
Thank you very much, Professor Farag. And
Yeah.
there is another question from, yeah. The question is from Mr. Asad. He's saying, why we prefer phenotyping over genotyping in case of metabolomics?
This would need no, this would need a long day to answer, my friend. But again, remember, we are at the end of we are the gene and our life experience. And gut microbiota could be one answer because your gene makeup definitely dedicate your gut residence. But again, your feeding behavior and your life experience, it's so complex. Genetics are indeed the main player, but it is very strange that such the small variance between humans, 0.3%, is what making all the differences as well. So it's a very complex story to tell. But what I came out to realize at the end, to be honest, that you have to consider all omics technology to really be sure that you are decoding the phenotype of your organism. This is but of course, we are also limited. The problem in these type of studies is that you need what we call an interdisciplinary research. You need the collaboration, you need resources. So yes.
Thank you very much, Professor Farag. And there is one opportunity that Comstech has recently opened, which is related to the field of food and functional sciences. So I would like to share that opportunity.
Sure, be our honor,
Yeah,
please.
yeah. So this is a Comstech Research Fellowship. I hope you can see, all of you can see my slide, my screen. So Comstech Research Fellowships for training in human nutrition, nutraceutical, and food sciences is open. And the deadline
Can you
for
put the link on the chat box, please,
yeah,
Hazima?
yes.
Thank you.
Yes, I'll put that link. And so I will request all the participants who are interested to please, apply using this link. And we hope that we can help you advance your careers and to link up with the expert scientists in the field, with the established laboratories. Professor Farag, I hope that on behalf of Comstech, we will be able to engage you further, seeing that the work that you're doing is very relevant and there would be many people who can benefit, and Comstech would definitely like to facilitate them and you to advance this area of research. With this, I would like to thank you for your lecture and for your time today. And
My pleasure.
and I would also like to thank our participants who joined us from different corners of the world. I can see people who joined from Africa, people who joined from Central Asia, and of course from Pakistan as well. Thank you very much for your time, and you all will receive your e-certificates for attending this webinar. And additionally, I see that there are a few people who are asking for the recording link. So this webinar is recorded and it will be put on Comstech's webpage or YouTube page, and you can access the recording from there. And with this,
Can you
thank
send
you.
me, Hazima, the recording, please, as well? The link would be great.
Yes, sir, I'll send that to you on the email.
Yes.
Because now they have to retrieve it and save it.
Yes.
So with this, thank you very much, everybody, and thank you, Professor Farag, for your time. It was truly an honor and an amazing experience to hear all of your science, and particularly in a very conventional way of presentation. Thank you very much. Thank you very
My
much.
honor.
Bye-bye. Thank you.
Bye-bye. Salam.
Bye-bye.
Salam for everyone. It was a pleasure to have you all, and thanks for your time, and all the best, dear colleagues. Take
Thank you
care.
very much. Thank you. Bye-bye.